Hello, welcome. Welcome to the BME seminar series by the Department of Biomedical Engineering at Columbia University. My name is Ed Guo. I'm the chair of the Department of Biomedical Engineering at Columbia. So today is a special edition of a BME seminar series. I will mention that. So which we continue to offer in hybrid format with both virtual and live audience. Our special guest speaker will present approximately 40 to 45 minutes, and we will leave you 15 minutes at the end for question and answers. So for our virtual audience throughout the presentation, please using the Q&A feature at the bottom of your screen, not the chat, to submit your questions. We'll try to answer as many questions as possible during the QA sessions at the end. For our live audience, you will have a chance to ask questions during the Q&A as well. And then I will pass in this microphone around such that asking questions. So today is really very exciting. This is our distinguished Van Simao lecture, named after our founding chair of biomechanics pioneer, Professor Van Simao. So a little bit history about Van Simao in this department. So Van moved from RPI in 1985 and joined as a faculty of mechanical engineering and also physics bioengineering at Columbia. And in December 24, 1995, you see, he's still working on Christmas Eve. And Executive Vice Provost Michael Crow walked into his office asking whether he'd be willing to establish a new department of biomedical engineering. So 1996, that's where we formed a Center for Biomedical Engineering, and 2000, the department was formed. So I think 2/3 of the faculty is recruited by Van Ma. And almost every single one of us still remain at Columbia. That says how wonderful department now. So our department now is more than have close to 25 faculties. And so Van is a pioneer, not only in the really changed way doing college biomechanics, bringing experiments and the theory, and really changing the world. So we are so glad have Celeste Nielsen, professor from Princeton University, really fitting to be giving the Van Simao lecture. So Celeste got a bachelor and a master from MIT, and they went to Johns Hopkins for PhD, which Johns Hopkins now we develop a very close relationship. We have our Rising Star workshop for underrepresented minorities. And then continued did a postdoc in Lawrence Livermore lab and joined Princeton's faculty ever since. So currently, Professor Nelson is a Wickham Family Professor in Bioengineering and Professor of Chemical and Biological Engineering and Molecular Biology at Princeton University. Her laboratory specializes in using engineering tissues, computation models to understand how mechanical forces direct development patterns events during the tissue morphogenesis, during disease progressions, with particular emphasis on the vertebrate lung. So her group is best known for revealing how mechanical forces from mesenchymes sculpt morphogenesis of epithelium. So when she's not thinking about the lungs, Celeste can be found running, planning next run, or daydreaming about tasting food to fuel his running. So for those people, if you follow my WeChat, I'm actually a very good cook. So hope to, in the future, be able to have Celeste enjoy my home cooking. And so it's such a pleasure. So let me presenting the Professor Nielsen, the Van Simao Lecture Medal. Thank you. So let me hold it.
Well, thank you so much for the wonderful introduction, as well as the invitation to be here today. This is an amazing department many old friends and new friends are here, and the leadership of Professor Mao, I think, is really appreciated throughout the entire biomedical engineering community. So it's a true honor to be able to be here today and to share my group's work with you. What I thought I would do is to share some of the work that we've been doing on, which is my favorite organ. I just love lungs, which is why I think about them all the time, even when I'm running in the morning, because they're so absolutely essential for our ability to survive. They're also really, I think, a beautiful example of this idea that the form of a tissue or an organ, the anatomy of that structure, is intimately coupled to its function, right? So you can remind yourselves of that fact by taking a deep breath. Go on, it's not quite lunchtime. Take a deep breath, right? And when you do that, you contract your diaphragm, which is this kidney bean-shaped sheet of skeletal muscle. You increase the volume of your chest cavity. That decreases the pressure surrounding the lungs and allows air to enter through the windpipe, the trachea, into the primary bronchi, and eventually to reach through all of the branches of this epithelial tree to eventually reach these cup-shaped surfaces known as alveoli, which are really, really thin and allow for oxygen to diffuse from the air side of this compartment into the bloodstream, and carbon dioxide to diffuse in the opposite direction, right? And so we're really interested in understanding how these structures build themselves for the purposes of eventually trying to replicate the construction of these tubes and alveoli, these tubes and cul-de-sacs, for tissue engineering purposes. So one question, or one way in order to address the question of how these structures are built, would be to carry out sort of very conventional developmental biology kinds of analyses that examine the signaling pathways that are activated as the tissues that are constructed within the lung, the airways and the alveoli, are formed during embryonic development. And that's an approach that has been pioneered by many luminaries in the fields of mouse developmental biology and genetics, and they've uncovered dozens of signaling pathways and hundreds of molecules that are activated and essential for formation of these tissue structures, some of which are shown here on this slide. But as an engineer, when thinking about all of these different molecules that have been identified, knowing this list of molecules doesn't tell us how the tissues themselves are constructed, right? It's sort of a parts list without the essential recipe for how to build those branches and those alveoli, those cul-de-sacs. So we've taken an orthogonal approach in my group, and that's to be purely agnostic to the molecules themselves and to envision or to treat the tissues as materials. Okay, so if you think of the tubes of your lung, they're lined by a single layer of cells. You can imagine that that layer of cells, that tissue, is a material, like the material that you see sort of forming the wall of a balloon, right? And if you want to take that simple material and turn it into a more complex structure, then you're going to need to know something about the properties of that material, whether it's elastic or viscoelastic, and then in response to forces being generated within the material itself or being applied to the material, you can take something that's as simple as a cylindrical balloon and turn it into the more complex structures that we see in the mature organ itself. Okay, so if you imagine then what it takes for the wall of that balloon, that straight material, to fold itself into a more complex architecture like you see in the trees that are within your lung, there are a few ways in which that material can fold itself into individual branches. Right? The first way is that the cells that line that material or that form that material can actively fold themselves into a new geometry, sort of like tissue origami. And then the second would be that the cells themselves, that material itself, is purely passive in response to forces being generated on it, either forces from the apical side, the side that faces the lumen, where air will eventually be after the baby is born, or it acts purely passively in response to forces being exerted from the basal side, the side that faces the mesenchyme or the stroma, and that will eventually be surrounded, in the case of the alveoli, by capillary beds. Right? So there's these different approaches in which you can imagine that a material is folded during development into the more complex structures that we see within our internal organs, including the lung. And so we've been using the vertebrate lung to try to uncover the mechanical forces that are important for building tubes and alveoli, and taking advantage of several different species, some of which are shown here, to really investigate sort of the anatomical, physiological, and mechanistic diversity that is present. One of the reasons why we have chosen this approach is that the conventional developmental biology kinds of approaches that have used genetic systems in order to uncover the parts list that I mentioned earlier have uncovered the exact same signaling pathways for all three different classes of vertebrates, despite the fact that the final architectures of these lungs look incredibly different. So that tells us that there's some interesting mechanics that's happening downstream of and perhaps differentiating these signaling pathways in the different organisms. So what I'm going to do today is to summarize a bunch of published work, first in birds, then in reptiles, and then in mice, and then tell you some unpublished work, an unpublished story, really focused on trying to use transcriptomics to uncover information about the mechanical forces that are involved in specifying tissue architecture during development. Okay, so our story starts with the humble chicken that we use as a model for development of the bird lung. We'll come back to why the bird lung is interesting at the very end, but its development begins as a simple wishbone-shaped structure within the lung that is lined by a single layer of epithelial cells, and branches form at very precise and stereotyped positions on the dorsal surface of this epithelium during development. And what we found is that, using this lung as a model system, that the branches that form on the dorsal surface of that embryonic epithelium do so actively. The epithelium itself is a highly columnar tissue that is comprised of basically really, really tall, really, really thin cells of rectangular cross-section. And the cells that are located within the tissue in the region that it will eventually form a branch undergo a change in shape, is an apical constriction, where they shrink their apical surfaces, which are adjacent to the lumen of the airway, and expand their basal surfaces, which are adjacent to the surrounding stroma or mesenchyme. And that change in shape locally of a subpopulation of cells is sufficient to exert enough force to push and bend the epithelial tube locally into a branch. We could model that process computationally by specifying the patterns of apical constriction that we observed experimentally on a thick-walled cylinder that we specified to have the mechanical properties, the material properties of the embryonic chick lung epithelium, and then applying the principle of volumetric growth. And so we see that local change in cell shape is sufficient to change the overall geometry of the tube itself. We went on then to find that those local bending of the epithelium, which pushes the tube into the surrounding tissue, is also sufficient to change the mechanical properties of the tissue that surrounds the epithelium. So specifically what we found is that the pushing of that epithelial branch into the surrounding mesenchyme causes a change in packing of the mesenchymal cells and actually causes them to become a little bit more extended, and they become more fluidic. They move around each other much more dramatically, much more dynamically than the mesenchymal cells that are located adjacent to the non-branching epithelium. And that actually decreases the resistance of the epithelium extending and elongating that branch into the mesenchyme. So in the case of the lungs of birds, using chick as our model, but also exploring ducks and quail, we found that the epithelium actually actively folds itself into new branches by this apical constriction mechanism. So what I can't tell you is what pinpoints the subpopulation of cells that are going to undergo this change in shape, but we're really interested in using apical constriction, local apical constrictions in sheets of cells to change the shapes of epithelia outside the body for engineering applications. Okay, so that's the bird, which has a really unique architecture of lung with respect to all the lungs that have evolved in invertebrates. We recently, right before the pandemic hit, got involved and really interested in trying to understand how the reptilian lung builds itself by using lizards as a model system. And one of the reasons why we were interested in investigating the morphogenesis of the lizard lung, the reptile lung, is because for many species of reptiles, especially lizards, the lung itself is fairly simple. It's basically a sac that has an undulating surface where gas exchange takes place. So those same signaling pathways that lead to the formation of branches in us and other mammals lead to the formation of an undulating surface in lizards. So we started by establishing the brown anole lizard as our model system. It's an invasive species that is prevalent throughout the South, especially in Florida, in Georgia. It's about the length of a pencil in terms of size of the adults, and can be bred rather easily in captivity. The eggs themselves, once they're laid, have a rather short incubation time of about a month, and the development of its lung is incredibly fast. So we established a colony of brown anole lizards and then mapped out development of the lungs. And so what we found is that the epithelium itself begins as a simple structure that looks kind of like sort of an uninflated water balloon. And so the epithelium is shown here in the top row and in green in the bottom row. At embryonic day 6 is the notation here in the bottom left, showing you one lung here. The epithelium is sort of like an uninflated water balloon. And then over the subsequent day, that epithelium goes from being smooth to being bumpy, having all these protrusions on the surface. At the same time as that epithelium is folding or being folded into these bumps, there's a second layer of tissue on the outside, on the basal surface of the epithelium, called smooth muscle. It starts out homogeneously covering the epithelium, so it's shown in the second row here and in purple in the bottom, at the starting point of lung development at embryonic day 6. And then as those bumps are forming in the epithelium, the smooth muscle actually migrates to form into a mesh, into a hexagonal mesh. And we see that the epithelial tissue forms protrusions then through the holes in the mesh. So what we have here is a system in which we have a simple epithelial sheet that is surrounded by a second layer of tissue. Morphogenesis of that second layer of tissue creates sort of a template, and then inflation, actually, by an increase in the volume of fluid within the lumen of this lung, pushes the epithelium into the holes in the lumen of the organ. So this physical mechanism could be predicted, again, computationally, by a model in which we have the epithelium shown here in green, the smooth muscle mesh shown in purple, and then a pressurized fluid within the lumen. Increasing the pressure, increasing the volume of the fluid within the lumen is, of course, as you might intuit, enough to push that epithelial tissue through the holes in the mesh. And so we call this process stress ball morphogenesis because the development of this lizard's lung, which takes place really, really quickly, is very similar to what happens when you squeeze one of those toys you might have on your desk. And we've been able to create engineered systems in which we basically increase the pressure within a model epithelium that we have surrounded with a smooth muscle-like mesh and been able to achieve the same change in shape and morphogenesis. So the reptile lung then builds itself using a completely different set of forces than the bird lung. We see no evidence for apical constriction within the epithelium of the lizard. Instead, what we see is a pressurization that takes place as water fills up the lumen of this developing lung. And that pressure is really essential for pushing the tissue through the holes in the mesh. If we disrupt the increase in pressure, these lungs don't form properly. If we disrupt the formation of the mesh, the lungs also don't form properly. Right, so completely different mechanisms here. Okay, so with that work as a backdrop, I want to spend the rest of the time telling you about some of our recent work, which has been focused on uncovering the mechanical forces that are essential for building the lungs of the mammal using the mouse as a model system. In the mouse, the lung begins as a simple tube, the trachea, which then splits in half to form the two primary bronchi that lead to the right and the left lung. And then the tips of those tubes branch recursively about 23 times, 23 generations, to build the final 8 million terminal ends of the airway epithelial tree that's present in the adult organ. Okay, so again, like in the lizard, we found no evidence for apical constriction in the formation of all of these branches within the developing mouse lung. Instead, what we found is a role for a second layer of tissue, the smooth muscle, which is forming around the airway epithelium at the same time as the epithelium is branching into the surrounding mesenchyme. And that smooth muscle differentiates from the undifferentiated mesenchymal cells that surround the epithelium during embryonic development. So we found a role for this tissue somewhat accidentally by carrying out time-lapse imaging analysis of embryonic lungs that were dissected from mice that label all of the smooth muscle cells within the developing organism in red via expression of red fluorescent protein. And so these mice express red fluorescent protein downstream of the alpha smooth muscle actin gene promoter. And what we found is that as branches form within the developing mouse lung, the smooth muscle is differentiating at the same time. And most importantly, in cases in which we have a tube that is going to split in half at its tip to form two daughter branches, we found that the site at which that tube is going to split is preceded by a spot of differentiation of smooth muscle cells right at the site. And so I hope you can appreciate that by looking at these time-lapse movies. They're a little pixelated, so I apologize for that. On the left-hand side, we have bright field images. Here's a primary bronchus going horizontally at the bottom. So the epithelium is this lighter color. There's a parent branch that comes out towards the top of the screen. It starts out looking somewhat like a thumb, and then its tip expands, so it starts to look like a mushroom. And then it clefts in the middle and forms a morphology that looks like Mickey Mouse ears. In the middle, we have the RFP signal. So this labels the smooth muscle cells and the cells that are differentiating into smooth muscle. They're wrapped around the outer surface of the primary bronchus. They're wrapped up the neck of that developing branch. And then I hope you can see that right before the epithelium undergoes this change in shape to form that Mickey Mouse ear morphology, there's a spot of fluorescence that appears and then brightens and expands. So we found, using a number of different approaches, that the differentiation pattern of that smooth muscle is actually essential for controlling the morphology of the developing epithelium. We think it's the stiffness of the smooth muscle that's important, and so we can also model this computationally using the same kinds of approaches. Here we have the epithelium in green, we have the undifferentiated mesenchyme in gray, and then we have the cells that are differentiating into a stiffer smooth muscle layer in purple. And as the epithelium grows and expands, because this is a highly proliferative tissue, the differentiation of a patch of stiffer cells at the tip of that structure is sufficient to guide the growth of that epithelium into that Mickey Mouse ear bifurcated morphology. So computationally, we find that to be the case. Experimentally, by disrupting smooth muscle differentiation, we also find that this pattern of differentiation really appears to physically sculpt the formation of branches within the developing mouse lung. And so in this case, what we think is happening is that the mechanical forces that are responsible for formation of these branches are really being templated by that stiffer layer of tissue that is surrounding the epithelium that is actively growing. And so the epithelium itself isn't actively folding, and instead it's growing and expanding, and its change in morphology that generates branches are being templated by, you could think of the equivalent of pushing forces from that stiffer tissue within the developing mesenchyme. So what we then wanted to do was to uncover what were the signaling pathways that specified the sites of smooth muscle differentiation. Because if we have 8 million branches within a mature lung, then we would need 8 million precisely timed, precisely positioned smooth muscle differentiation events. So that's where we started, but as we were carrying out that analysis, we were somewhat thrown for a loop. This was right as the pandemic was starting, because a paper came out from Shen Sun's group at the University of San Diego, where they concluded that smooth muscle differentiation is dispensable for branching of the epithelium. And so they came to this conclusion based on results from a genetic model where they inducibly floxed out, so inducibly deleted myocardium, a key muscle transcription factor, from the developing mouse lung mesenchyme using a lung mesenchyme specific TBX4 RTTA TET-on-CRE driver. So this driver means that in their genetic crosses, myocardium is deleted only from the mesenchyme within the developing lung. Okay? When they did this, they found that this depleted the presence of alpha smooth muscle actin expressing cells around the airways. So you can see this purple color in their control that goes around the green airways, and it's missing in their knockout mouse. They also found that deleting myocardium from the mesenchyme of the developing lung led to decreases in the expression of the transcript for alpha smooth muscle actin, which is also known as ACTA2, and decreased the expression of another smooth muscle marker called transgelin. And they found, somewhat surprisingly to us, that when they carried out this deletion, which they concluded completely got rid of smooth muscle, that the lungs formed the exact same number of branches as controls. Okay? So their conclusion was that smooth muscle was not needed for formation of branches within the embryonic mouse lung, which was the complete opposite, of course, to everything that we had been uncovering using our engineering and mechanical experimental approaches. So we were both confused, a little distressed, confused, but also somewhat excited, after you sleep on it for a little bit, somewhat excited by this paper because it suggested that there was more going on within that developing tissue than any of us had possibly imagined. And we also thought that really understanding this morphology and the morphogenesis of this system could tell us something more about the mechanical forces that were important for building the mammalian lung. Okay, so we asked, what's going on here? Well, what we wanted to do was to really dive in and analyze that genetic system a bit more. Unfortunately, we were unable to obtain the myocardium floxed mouse. But as you might be aware, myocardium doesn't act on its own. It's actually a cofactor for a much more important transcription factor called SRF, or serum response factor. These two proteins together activate the expression of smooth muscle genes. And so you need both. Neither of them is sufficient. And so we had the SRF floxed animals, SRF floxed mice, cryo-recovered from Jackson Labs, and started to establish a colony at Princeton so that we could carry out a similar analysis and then really understand mechanically what was going on in the development of these airways. So this was work that was carried out by our most recently graduated student, whose name is cut off here. Her name is Katie Goodwin, She just defended her thesis a couple of weeks ago. We are very sad to see her go, but very excited for what she's going to do in the future. She's awesome. When she starts applying for faculty positions, everyone should be on the lookout for her because she is going to be a massive superstar. She's already a superstar. Okay, so Katie set up a parallel system to what the Sun Lab had developed using the exact same Cre driver. So again, TBX4 RTTA tet on Cre is only expressed in the developing lung mesenchyme. She used that driver to delete SRF from embryonic mice and then carried out the similar analysis to what had been published by the Sun Lab in 2020. So she's first examined the expression of alpha smooth muscle actin as a marker for smooth muscle cells in her controls as well as in her knockout animals, and she found that just as the Sun Lab had reported, getting rid of SRF from the mesenchyme within the developing lung blocks the formation of cells that express alpha smooth muscle actin around the airways. So you see an absence of purple color around the green epithelium here. She also found, in comparing the numbers of branches that form in the knockout animals versus the littermate controls, that they form the same number of branches. And so this deletion phenocopies the deletion of myocardin from the developing embryonic lung mesenchyme. So then what Katie did was to take the analysis one step further. She wanted to know what those cells were. So what is deleting this really important transcription factor doing in the developing lung? And so in order to carry out and really address that question, she conducted a single-cell RNA sequencing analysis of both the wild type and the SRF knockout embryonic lungs. And what she found is that the lungs show the exact same cell types, the exact same clusters, regardless of SRF expression. So we have epithelium, mesothelium, vascular endothelium, neurons, immune cells in both the wild type and the knockout animals. And then we have this very, very large population of clusters of mesenchymal cells that are highly related to each other that include smooth muscle, as you'll see in the case of the control. So what she then did was to confirm that deleting SRF actually had the intended effects by looking at the expression of markers within these UMAPs of the single-cell RNA sequencing data. So on the top we have the control lungs. They express alpha smooth muscle actin. That's that dark purple color towards like 5 o'clock in this mesenchymal cluster. And they also express transgelin in that same region of the UMAP, in that same population of cells. Deleting SRF gets rid of alpha smooth muscle actin, ACTA2 expression within the mutant mesenchymal population, mesenchymal clusters, and also gets rid of the expression of transgelin, similar to the PCR results that the Sun Lab had reported. Similarly, Katie found that deleting SRF using this TBX4 RTTA tet on Cre driver disrupted the expression of SRF in the mutant compared to the control. So in the control, you can see that SRF is lighting up the entire mesenchymal cluster. So you have that dark color throughout that cluster of cells. It's pretty much absent within the mutant, telling us that our Cre was effective. What was surprising, though, was that we found that we still had a subpopulation of cells in the mesenchyme that expressed the cofactor that really marks smooth muscle, the cofactor myocardin. So in the control, we have this population of cells in the same position that expressed alpha smooth muscle actin and transgelin, also expressing myocardin, so in this 5 o'clock position. In the mutant, we also have a cluster of cells that express myocardin. So despite SRF being deleted, we have myocardin expression. And those myocardin positive cells that lacked alpha smooth muscle actin were actually located in the same place as smooth muscle cells. So we found this by carrying out RNA scope analysis. So it's basically an in situ hybridization for the transcript of these genes, which are lighting up here. So in the control, this center tube would be the airway epithelium. It's surrounded by alpha smooth muscle actin, so ACTA2, labeling in the same position as we have myocardin labeling. In the mutant, we have 2 airways down here. ACTA2 is missing, so there's no alpha smooth muscle actin, but we have myocardin expressing cells wrapping around the airways. And importantly, Katie found that these SRF mutants still express non-SRF targets that are typically markers of smooth muscle. So these include HIP, SMOC2, IGF-1, and TGF-bI in that smooth muscle population in the control. They're expressed in those myocardium-expressing populations of cells within the mutant. Right? Katie then carried out a KEGG pathway enrichment analysis to see which pathways were activated within the control and the SRF mutant smooth muscle clusters. And she found most of the exact same signaling pathways are activated in these two populations of cells with two really notable differences. In the control, we have pathways that are associated with muscle contraction. And in the mutant, we have a massive overrepresentation of pathways associated with pluripotency. Nonetheless, they're very similar to each other. So Katie then surmised that deleting SRF signaling might still permit the differentiation of smooth muscle, but maybe not the expression of all of the markers of smooth muscle, and that SRF null population might still have the same or similar mechanical properties as the wild-type tissue. So to test that second hypothesis, Katie carried out one of these dissection experiments. They're actually kind of cool. She took a lung out of an embryonic mouse and then waited until there was a time point in which the epithelium had formed that Mickey Mouse geometry and dissected away the mesenchyme and the smooth muscle. And because of how the tissue deforms mechanically, removing that constraint allows the epithelium to pop back into sort of a mushroom geometry. So pay attention to the before and the after. That's probably the most important portions of this analysis. She did the same thing for the mutant and found that in the SRF mutant lungs, they also pop back from a Mickey Mouse to a mushroom geometry, telling us that those mechanical attributes of the mesenchyme are the same within these tissues. So from these analyses, we concluded that disrupting SRF or myocardium signaling does not eliminate airway smooth muscle per se. We still have this population of cells that's missing SRF, missing ACTA2, missing a few other markers of smooth muscle, but is otherwise identical to the wild-type population. So we think that the correct interpretation of this paper is not that smooth muscle is dispensable for epithelial branching, but that the gene myocardium is dispensable for epithelial branching. And that getting rid of myocardium from the mesenchyme does not completely delete or deplete the mesenchyme of airway smooth muscle. All right, so what is this SRF or myocardium null population? I see Ed nodding his head over here. So those of you who study the vascular system, for instance, will know that smooth muscle doesn't exist in one phenotype, but it actually transitions between several phenotypes, two of which are shown here. So there's contractile smooth muscle, which expresses ACTA2 and transgelin and other genes associated with contraction. That can transition to a more proliferative or synthetic phenotype in cases of disease and situations in which one needs repair, for instance. They transition between each other in response to changes within the surrounding microenvironment. So Katie surmised that SRF myocardium signaling might actually be important for the switch between these two fates. She compiled a list of markers associated with the synthetic proliferative phenotype or the contractile phenotype, shown in pink and gray respectively. And then she clustered her smooth muscle cells from the control embryonic lungs and those from the SRF null embryonic lungs, and found that the control smooth muscle expressed more markers of contractile phenotype, and the SRF null smooth muscle expressed more markers of synthetic phenotype. One of those markers was the gene collagen 2A1, which Katie found using RNA scope analysis is actually expressed within the mesenchyme adjacent to the epithelium in the developing mouse lung. So she confirmed that it's in the correct location within these developing systems. Another of these synthetic proliferative markers were genes associated with the cell cycle. So Katie carried out proliferation analysis by either looking at Ki67 staining or carrying out analysis of the phase of the cell cycle of her single-cell RNA-seq data. And she found that the SRF null population was actually more proliferative than the control. So we conclude that this SRF null smooth muscle actually exhibits what's probably been thought to be mostly an adult phenomenon of a classical synthetic/proliferative phenotype, suggesting that cells undergo these fate switches in the embryo as well. So our interpretation then still stands that the growing airway epithelium is really responding to the patterns of stiffness within the surrounding mesenchyme. Those patterns of stiffness are dictated by the differentiation of smooth muscle cells, and they actually arise fairly early in the differentiation pathway. We don't need contractility of the cells. We don't need expression of the contractile portions of their proteome. All we need is really the baseline levels of expression of smooth muscle markers. And that, of course, to remind you, is quite distinct from the mechanisms that build the bird lung or the inflation mechanisms that build the reptile lung. All right, so in the last couple of minutes, why aren't these mechanisms conserved? Right? I didn't show you the data, but I alluded to the fact that the exact same signaling pathways have been identified in all three classes of vertebrates. And yet we end up with three very different physical mechanisms for building the lungs. This is a conundrum, right? It makes many of us uncomfortable. We like for things to be conserved, and this is not conserved. I think it's beautiful. Diversity is beautiful. Diversity of mechanism is beautiful. So this is pure speculation. Speculation alert. We have no evidence for this, but we're purely speculating. Okay, so I introduced the problem to you by introducing the analogy to materials, like a balloon. And I said that the properties of those materials are going to be important for specifying how they respond to mechanical forces, right? Well, it turns out that the properties of the epithelia of these three different classes of vertebrates are very different. So to go back to our balloons, you can think of it as like, it's really easy for many of us, not all of us, to blow up a regular balloon. But if you've ever bought one of those punch ball balloons for your kids that have the really, really thick walls, those are really hard to blow up. They're hard for us to inflate because of the property of the wall of the balloon, right? So we find that there are differences in the property of the walls of the epithelia within these developing vertebrates as well. So the bird embryonic airway epithelium is actually highly columnar. The cells are about 2 microns wide, but they're like almost 30 microns tall. So they're really, really high aspect ratio. The developing lizard has initially a cuboidal epithelium, so the cells look rather rectangular. And then as those inflate, they become squamous, so really, really flat. The mouse has a highly proliferative pseudostratified epithelium. So the cells within this tissue are going all sorts of different directions, very different morphology than what we see in the reptile or in the bird. So we have divergent initial conditions here, right? Very, very different tissue structures. In the case of the bird, this tissue, at least from the kinds of tests we've been able to do, is both thick as well as rather rigid. It's hard, to go back to the balloon analogy, it's hard for us to blow it up using pressure. So it folds itself because, as you might imagine, the amount of force necessary to be directed on the luminal side or the basal side is probably large in order to build a branch here. In contrast, both the reptile, the lizard, and the mouse have thin, floppy epithelia that expand, either because of fluid pressure within the lumen here or because of being highly proliferative in the case of the mouse. And so these two tissues actually respond to forces that are being exerted by their surroundings. I also said that we get our final structure from folding those initial starting materials. And here I think it's quite revealing that the final conditions across these three classes of vertebrate lungs are also very different. So when we breathe, when you contracted your diaphragm, when you took that deep breath, we actually change the volume of our lungs, right? Our lungs are floppy. They're compliant. They inflate and they deflate. That's how we breathe. Lizards are the same. Birds have thick, rigid tubes. Their lungs don't inflate or deflate. They're thick and rigid and completely confined to the same volume through the entire breath cycle. And instead, they push air through these rigid tubes by a second tissue structure called an air sac that undergoes the contraction. Okay? So we have different initial conditions, different final conditions, different anatomies, different physiologies, and so it should probably not be surprising that we have different mechanical forces that get us there. So we are continuing to really build up this zoo that we've got going in the lab to try to uncover other developmental motifs. We've recently started working on the veiled chameleon. We have experiments being carried out with a marsupial species. We have a bunch of other reptiles as well as amphibians that we're looking at. We really want to know what controls patterning within these contexts, and how much of those patterning mechanisms can we actually actuate in engineered settings to take advantage of evolution's diversity to build new tissue structures. So all of the work that I showed you was carried out by a really fantastic team. The unpublished work by the recently graduated Katie Goodwin, who's truly fantastic. They have been outstanding through all of the ups and downs of the last few years. And with that, thank you so much for the opportunity to be here, to share with you today, and for your attention through these different stories of different critters. And if there's any time, I would be really, really happy to take any questions. So thank you.
Yeah, thanks so much, Celeste. Fantastic. And you see, our student prefers sitting on the floor instead of sitting next to me. So if we, okay, we're done that already. So before we go into question and answer, I would like to tell everyone attention to next hybrid seminar, which will taking place next Friday, October 7th. We'll hear from our own Professor Sonia Vikovich as she presents spatial omics technology at scale, and will be not in this room, and we'll be back to our regular classroom. And then please follow us on social media to receive the latest updates. You can see our social media information on the slides, and you can always access the registration information for our BME seminar on website at bme.columbia.edu. Now I'm passing to Professor Olsen for handling the questions.
Great. So we will take some questions from our virtual audience and also from you guys. So, but I'll start with you. Any questions from the students or anyone who's attending here?
Thanks, Celeste. Really beautiful talk, as always. You told us about kind of differences between kind of potential physical mechanisms in birds and mammals and reptiles. I was wondering if, say, even within birds, are there interesting scaling problems, say, between a hummingbird and an eagle and kind of the final branching structure, length scales, and is there anything known about how that's solved developmentally?
Okay, wow. That's an awesome question, Karen. So we have not thought about that problem in the context of birds. There are certainly scaling issues. As far as we've learned, the mechanisms appear to be rather similar across the bird species we've been able to examine with respect to the initial formation of branches, which takes place by the active folding apical constriction event. I think where the scaling will become interesting, actually, in the case of birds, is in the very last stages of development of the lung. So there they have this really beautiful event that takes place where all of those branches that have formed have to extend over the entire distance of that final organ, which is centimeters in chickens, and it must be much, much larger than it's about a centimeter in birds, much, much larger than a centimeter in the case of an eagle. But anyway, those branches have to extend across the entire length of the organ and then find their partner and fuse. And so you have this really beautiful path-finding and then epithelial anastomosis that has to take place. We know physically how that fusion takes place in chicks. We know nothing about how the epithelium is able to find its partner over those long distances. And so as you increase the distance, I think that the question that you ask is going to become really, really important to understand the answer to. Because it would reflect probably something that can scale across length and also time, as some of these birds have longer gestations, or longer incubation times. Yeah.
Hi, thank you so much for the presentation. I wanted to ask if you've done research on amphibians already, and or what do you expect to be different with the amphibian lung development, because the amount of time they spend in water is different from a mouse and all the birds you've talked about.
Wonderful question. We are waiting for IACUC approval to get tadpoles into our facility. We were literally having a conversation with them yesterday. So we are planning to look at amphibians, in part because they form lungs during metamorphosis. So the tadpole forms the lung as it becomes a frog. So imagine going through puberty. This is like the equivalent to me. Imagine going through puberty and developing an entirely new internal organ. That's what these tadpoles are doing. And what's really cool about development of the lung in an amphibian is that, at least in the case of the laboratory model amphibian species like Xenopus, Laevis, or Tropicalis, the lungs are actually being used as they're developing. So the little tadpole, after a point in its metamorphosis, will go to the surface of the water and take a gulp of air, and that air will go into the lungs. And so now we have completely different boundary conditions in the case of development of the epithelium in an amphibian as compared to a reptile, a mammal, or a bird. Right? So while our lungs are developing, they're filled with aqueous fluid. The same is true for the bird. The same is true for the reptile. Here we have, in the development of an amphibian, the lung being full of air. So when you change the boundary condition, just like when you change the initial condition, the mechanics have to change, right? So we are almost to the point where we've got tadpoles on campus in order to investigate this, just waiting on final approval. That's where I think the differences are probably going to lie, is how do you apply force in the context of an air-tissue interface sufficient to allow the tissue to change structure?
Okay, maybe we'll take one question from your virtual audience. So you're getting a lot of questions, and later you can do these that we don't get to. But this one sounds nice, from Jessica T.O. Great talk. I'm curious if there's a particular lung morphology, tissue dynamics that is most efficient in gaseous exchange, considering the diversity of physical mechanisms you've successfully dissected.
There is. So the question is about the efficiency of gas exchange. So generally speaking, if you just evaluate these three different systems from the engineering perspective, the bird lung is the most efficient gas exchanger. Because they have this continuous flow of air through those airways that have, fused during that second half of development that we just discussed. So you have continuous flow of air, and then the capillaries are organized, so if air flows this way, the capillaries are organized perpendicular to those tubes. And so you have here a cross-current gas exchanger, which is the most efficient gas exchanger you can design from the engineering perspective. You also don't have any mixing of fresh air and stale air from that unidirectional flow. And so purely from the design perspective, they have the most efficient design. That being said, there are tweaks you can make to the mammalian design to make it more efficient. Right, so if you decrease the thickness of the epithelium within the alveoli, that decreases the barrier to gas exchange. and there are certain species that have accomplished that quite well. If you increase the concentration of red blood cells, for instance, flowing through the bloodstream, or increase the concentration of hemoglobin within those red blood cells, then you can also increase the driving force for gas exchange. So there are tweaks that various animals have made to get over the rather inefficient design that we have for the mammalian lung. But from the pure engineering perspective, the bird system is the most efficient.
I remember years ago reading about or hearing a talk about a goose that flies over the Himalayas and all the different things that they do in order to get better exchange. So really cool. Anyway, let's. I know that we're done. I have a question here, so let's let her speak here. Hi, Celeste. Fantastic talk. Do you know if these diverse factors involved in lung development also regulate the pathological changes in lung structure and function, for example, during lung fibrosis?
Insightful question. We don't know. To be perfectly honest and blunt, we have no idea. And I think there we have to really start to understand that coupling between conserved signaling pathways, conserved gene expression patterns, and divergent mechanical systems. And until we can understand how the same signaling pathways give you very different, mechanical force outputs, we won't be able to uncover the answer to Gordana's question with respect to what happens later on during disease. So unfortunately, we don't know.
Okay. So maybe if we have one more question from the audience, and in the very back, or no, there's one.
Okay. Wow.
Does amniotic fluid involve physically or molecular level development?
Yeah.
I love this question.
Yeah, the question is whether embryonic fluids have anything physically or molecular mechanisms influence
Yes.
this.
So, absolutely. This is the sort of the direction that my lab is heading in the future, actually, is to really understand the role of mechanical signaling from that compartment. So just to remind everybody, when you're developing as a fetus, you're developing in the uterus, and you are surrounded by amniotic fluid. That amniotic fluid is actually continuous with the fluid that is within the airways of the lung. The composition of that fluid has not been monitored over developmental time, like to the level that we would need in order to understand the potential role of chemical signaling. But we know that changes in the pressure of that fluid in the second half of gestation are really critical for determining the extent to which the lungs develop at an appropriate rate. And similarly, alterations within the pressure of that fluid or the volume of that fluid can have serious consequences for development of the fetal lung. So absolutely essential, and we hope to have answers for what that's doing, what that fluid is doing, in the next 5 to 10 years. So thank you for the question.
So I think we should thank our speaker one more time.
Thank you so much.
Thank you so much. That's wonderful.